I'd be happy to relay your questions to our panelists and get you involved in this conversation. So, the year 2020 was intended to be a milestone year for HIV and AIDS, as organizations like the American Foundation for AIDS Research had aimed to find a cure by now. Unfortunately, we have a long way to go, but groups like UNAIDS and International AIDS Society have made it a point to rebuild the momentum around the AIDS epidemic by focusing on what still needs to be done. Our discussion today will focus on where we are now with AIDS and HIV research, how the current pandemic impacts global response, and the goals that still need to be met to make a difference. How the current pandemic impacts global response. I think we have an echo, but I'm going to continue going to make a difference. All right, so I'm Alexa Record, a media and events coordinator at QBI, and today we have with us Jeffrey Johnson and David Gordon. Could you two introduce yourselves and briefly tell us about your general expertise? And today we have with us Jeffrey.
Sure. Is there a little bit of feedback? The YouTube window open maybe? Sure. Is there a little bit of feedback? The YouTube window open maybe? Ah, there we go.
Oh my gosh. Okay, so hopefully that wasn't too distracting.
That's okay. My name's Jeff Johnson. I'm an associate professor of microbiology at the Icahn School of Medicine at Mount Sinai in New York City. And I study how HIV takes over the cells that it infects.
And I'm David Gordon. I'm an associate researcher and rising assistant professor at UCSF in the lab of Nevin Krogan. And I work on more molecular details of how HIV genetically changes and hijacks the cells that it infects.
Great, thanks guys. So to put everyone on the same page, can you tell us what HIV and AIDS is and maybe point out the differences between them?
Sure. So HIV is the virus, human immunodeficiency virus, and AIDS is a disease that can result from HIV infection. AIDS is usually defined by a CD4 T cell count. So it's defined by how healthy your immune system is, but it is the disease. In the same way that SARS-CoV-2 is a virus and COVID-19 is a disease, HIV is a virus, AIDS is a disease that results from infection.
Great. Okay. So I want to talk about your insights on the history and the future of HIV response, personally and scientifically. So tell us a bit about how you each got involved in working with HIV. What keeps you interested and why it matters to you? Jeff, do you want to start?
Sure. So I'm gay, and so HIV affects my community in a pretty profound way. There are people who I know and love who are affected by HIV, who are living with HIV. I didn't exactly set out to study HIV as a scientific career. I was interested in infectious diseases and a technique called mass spectrometry. It just so happened that when I was at a job I wasn't very happy with, there was a job that I applied for studying HIV, which was at UCSF for Nevin Krogan, who is now the head of QBI. And so really it comes down to the federal government had quite a lot of funding available for HIV, which is how Nevin got involved in studying HIV and had a job opening available that I ended up applying for. So it's sort of a combination of a number of factors that boils down to sort of the social response to HIV as a disease.
And I got involved as a postdoc as well. And so I began my career as a cell biologist. I studied the molecular underpinnings of life, essentially, how our individual cells function. And then I moved to Nevin's lab, and I wanted to study something called a genetic interaction. And basically that means how two functions of two genes impact on each other, whether they might function doing the same thing in the same molecular machine or maybe do things in parallel, different molecular machines. And when I joined the lab, I really wanted to study that technique. And Nevin told me, well, if you want to do that technique, you should really do it with HIV. We've just generated this entire interaction map of how HIV hijacks human cells and what human proteins HIV is binding to. And wouldn't it be great to look at how those human proteins, how their functions impact on each other in mediating HIV infection? And that's how I began my studies of HIV, which have then led in actually several different directions subsequently.
Great. So some say that the scientific community has developed a new understanding of the challenges that must be overcome to get a cure. So do you agree with that? And do you feel that scientists are more equipped now than they were when the AIDS outbreak began, or even from when you started working with HIV?
We're certainly better equipped now than we ever have been. The advent of RNA interference, which allows us to deplete certain proteins or gene functions from the cell, as well as CRISPR in the last 7 years, which allows us to completely eliminate genes or introduce new genes or mutate things in the genome. These new genetic tools have been a real revolution in how we study any number of biological questions, and definitely also for HIV infection. So that's allowing us to understand the virus better. And in understanding the virus better, we can understand how to combat it better.
Yeah, I think, I mean, compared to the beginning of the epidemic when there were no treatments, I think HIV is one of the great success stories of science. You know, it took some time, but eventually now we have really good, really effective drugs that can turn a very deadly disease into what's essentially a chronic illness. It's not perfect, it's not a cure, but it allows us to treat individuals so that they can live fairly normal lives. So I think the barrier now is to getting a cure. You know, HIV is a very insidious virus. It actually integrates into the DNA of our cells. And once it's in there, it's very hard to get it out. So a lot of people who focus on curing HIV, they're trying to get these cells who have HIV integrated into their DNA to turn back into infected cells so that we can kill them. I think there are a number of recent cases of people who have been cured of HIV. You know, there's the Berlin patient, the London patient. These are individuals who had cancer. They had bone marrow transplants where they gave them bone marrow from someone who's resistant to HIV. They have a mutation, and they're essentially cured. In one case, they actually reverted back, and the virus came back. And then more recently, there's a Sao Paulo patient who I think is really interesting, although it's pretty early. They did not get the bone marrow transplant. They had a nicotinamide treatment, which is a drug that they use to sort of wake up latent HIV in that individual, and then killed off all of the HIV. And that person is also now, as far as we know, free of HIV in their system. Of course, it'll take some time to know if they're truly free of HIV. It can hide in certain tissues and can be very difficult to detect. But there's a lot of exciting movement there. You know, the idea of anyone being cured of HIV seemed like a long shot in science for a long time. So just seeing that it's possible, and then, you know, building on these findings, and like Dave said, understanding the basic biology of how the virus takes over the cell, how it goes into the DNA, and how do you reverse those processes. We know a lot more, and it's an exciting time to study HIV.
No, definitely. So you mentioned these 2 patients that scientists have been able to cure. Do you think that scientists have tried to replicate that? Is it by chance? It's been years.
Yeah, I don't know if people are still trying to do bone marrow transplants. I know for the Sao Paulo patient, that was part of a cohort, I believe, where they tried it with multiple patients, and one of those patients, they successfully, quote unquote, cured them of HIV. That was just presented at the AIDS 2020 meeting earlier this year. So it's, yeah, I don't know. I know that people are trying to cure HIV. I don't know what's happening in terms of clinical trials to do that and how successful they are. I think as soon as they're successful, you'll hear about them. So,
right, I'm sure. So what I hear from you both is that we've had a tremendous amount of progress, but there's still work to be done. Before we move on, Dave, do you have anything to reiterate what the typical roadblocks to a cure are?
Well, in terms of roadblocks to a cure, as Jeff mentioned, there's this concept of reawakening the HIV, and because HIV integrates into our very foundations of our cells, the DNA, that it integrates into our genetic code, and so it's not a trivial thing to get rid of such an event. You know, it's now built into the building blocks of some of our most long-lived cells, and so there's two ways you can imagine eliminating such a virus. You can either reawaken all of it, and then in doing so, attack it either with the immune system or somehow get those cells to die. Or there's another approach, which people call block and lock, where you simply silence all of the HIV that's floating around in these reservoirs in the body. And so both have their challenges. Of course, the case that Jeff mentioned, this nicotinamide patient, that's extremely exciting for the field. But I think time will tell exactly whether that lasts. I think other similar treatments have actually, we've been researching this for a long time, how to either silence the virus or reactivate it. And I think that line of investigation is something that needs to continue, and I think is happening in many labs throughout the world.
Great. Yeah. So let's talk about the links between HIV and SARS-CoV-2. So 2020, this year, a lot of the attention has gone towards dealing with the containment of COVID-19. Do you think that strategies used to advance HIV and AIDS research could advise how to handle research on COVID-19 and vice versa?
Absolutely. So speaking just from our group, years ago, Nevin's group published a study looking at the HIV interactions with human cells, mapping all of them, hundreds of them. More recently, I led an effort in a group to then do the same thing with SARS-CoV-2. Actually, before it was even called SARS-CoV-2, we went ahead and took every single protein from SARS-CoV-2, the causative virus for COVID disease, and we looked at every single human protein that those proteins bind to. And we built a big map and we put it online in March. And it's been fueling an immense amount of other research, including the identification of a few drug targets, which we validated with our collaborators in Paris and in New York, which are under now further investigation. And so I think that's just one example of how certain techniques that we originally pioneered in studying HIV can then be rapidly repurposed for studying other viruses. In terms of repurposing other antivirals as well, some of the first antivirals that were tried on SARS-CoV-2 were HIV protease inhibitors. Those were ultimately not found to be very effective. However, it's still interesting to note that antivirals that work on one virus sometimes can be used in another, and there are several examples of clinically approved antivirals that are used in several viruses.
Well, is it true that remdesivir was developed first as an HIV antiretroviral? Is that right? I thought I heard.
I believe remdesivir was tested on Ebola most recently. And so it inhibits the RNA-dependent RNA polymerase of SARS-CoV-2, and it's an effective treatment. So exactly, Jeff, like another example of repurposing. And not to go too far with this, but we can repurpose not only drugs targeted towards the virus like remdesivir, but also drugs targeted towards human proteins. But that's another conversation.
Yeah, I think something really interesting about HIV is we've been studying it very intensely for the past 40 or so years, and we've actually begun using HIV as the tool. We know so much about it, and it integrates into the genome of whatever cells it infects. So you can use it as a tool in the laboratory if you need to, for whatever research or therapeutic purposes. If you need to deliver a gene into a cell, we use HIV as a backbone to do that. So we use it very frequently to modify cells, to modify their genomes. So it's not just that the research that we learn about HIV applies to the SARS-CoV-2 basic knowledge, but it's also, the more we know about these viruses, we can use them for ourselves, re-engineer them, make them safer to use them to deliver therapies or to modify cells in the lab in order to research something else. It's a really useful virus in many different ways, surprisingly.
Right. So while we're on this topic, we have an audience question that's related. How has the research on HIV impacted SARS research? You touched on it, but is there anything else to add?
I mean, I would say when I started at UCSF working for Nevin Krogan, that was 11 years ago. Makes me feel a little old. And I joined his lab to do HIV research. And at the time, he didn't have, again, I'm talking about this technology, mass spectrometry. He didn't have a mass spectrometry lab within his own lab. So he hired me to build this mass spectrometry lab. And HIV was the first thing that we were studying there. So everything we developed in that lab for mass spectrometry was using HIV as the application. So we developed ways to find how proteins interact with each other, or how proteins are modified during infection, or where proteins go within the cell, things like that. All of that was developed for HIV as sort of the base, and then we started repurposing what we had built for other viruses. So as soon as SARS-CoV-2 came along, all of those tools were ready to go, and we could very quickly apply them to SARS-CoV-2. And it was like really an astounding turnaround between when the virus was published online, the genome sequence, and when these papers came out with, Dave's first author protein interaction paper, and then a post-translational modification paper that also came out of Nevin's lab. We would not have been able to do that as quickly if we hadn't started everything that we did using HIV.
It's true, yeah. And in terms of just generally trying to repurpose not just HIV antivirals, but other antivirals, the more we know about viruses have an evolutionary tree. Just like we're related to monkeys, viruses can be related to other viruses. And if you have antivirals that work on one, oftentimes, sometimes, they can work on another. And so the comparison of HIV and SARS, unfortunately, the HIV protease inhibitors don't really work on SARS. But for example, this Ebola antiviral remdesivir does work on SARS. And now Gilead is making buckets of it to treat numbers like hundreds of thousands, millions of patients could and hopefully will be treated successfully with drugs like that. At least if there's not enough time to make a new antiviral, let's see what we have on the shelf. And so I guess this whole webinar is about how do we looking through the lens of HIV, but also more broadly about antivirals and how they can be used to target emerging threats, I think is another important thing to touch upon.
Mm-hmm. So it's encouraging to hear how one virus can inform the other. But while many people pay attention to COVID-19, how can we make sure not to drop the ball on HIV and AIDS epidemic? Can we do both?
It's difficult, I'll say. Resources are finite. And so, at least in my personal experience, I moved to New York City about a year ago to start my own lab here at Mount Sinai. And then, of course, a pandemic comes along. And in December, by March, it was in New York City, and we had to shut down all of our labs. And the only research that we were allowed to continue doing was COVID research. We couldn't do HIV research. We had to set everything aside. And not just HIV, that's everything. That's cardiovascular disease research. That's neurodegenerative disease research. Everything has to get set aside until we can solve this problem. And so a lot of resources right now are dedicated to studying SARS-CoV-2 and COVID. Again, there's just finite resources. You can't do everything all at once. And so the sooner that we can get past this pandemic with masks, social distancing, a vaccine when it becomes available, the sooner we can get over this, the sooner we can get back to studying the diseases that have been impacting us for a very long time.
Yeah, just exactly right. So in the lab, we face the same challenges everybody faces in their own in broader society. We need to social distance. We need to put in new standards and uh, protocols in place to keep ourselves away from each other, to have masks on. And what do you do, for example, when you need to train someone new? Well, you have to wear a really effective mask, and you have to try to maintain social distance, but also guide them in how they do their research. So I think the practical matters of just how you do research under social distancing is a real challenge. In the context of HIV, there's still hundreds of thousands of people who will likely die of HIV and AIDS-related causes this year. And so it's important to continue that research. It's important to continue research into other diseases as well. However, we do have to be careful that our colleagues are safe. And so yeah, I think a lot of us since March have had to come to grips with the practicalities of how to do research in this new environment. And like Jeff said, wear masks and enforce social distancing as much as possible. And I think it's up to all of us to try and help get this under control as a society.
Right. I'm glad you bring this up. It sounds like what I'm hearing is COVID-19 has affected people with other health issues, and not many people are talking about it. And perhaps, social distancing and government lockdowns have disrupted HIV prevention, treatment efforts, and even maybe HIV research. So kind of to that topic, we have another audience question. Are people living with AIDS more susceptible to COVID-19?
I think actually no. So I've seen a report. I don't think it's a peer-reviewed publication, but I've seen a report that said that people living with HIV have a slightly lower risk of developing COVID-19, which might be a result of them being on antiretroviral therapies that may have some limited effectiveness against COVID-19. Again, I don't think that's peer-reviewed. That's just a report that I saw in one of the HIV magazines. It wouldn't surprise me. Like Dave was saying before, oftentimes antiviral medications target or are effective against more than one virus. But again, I think it also depends on how far along AIDS progression is. If you're, a late stage AIDS patient with, completely dysfunctional immune system, then you may have a much worse outcome with COVID-19. At the same time, COVID-19 severe disease is associated with an overactive immune system. Usually it's the inflammation, the immune cell infiltration, that's what causes those really severe, acute respiratory distress, the ARDS, whatever that stands for. And so, but in general, no, I think the answer is they're not more susceptible if not more protected.
Yeah, Jeff's exactly right. And I think it's just building on his comment, that you have this, what they call cytokine storm, cytokines being these chemical messengers that trigger inflammation in this ARDS, this acute respiratory distress. And so one of the drugs that has been found to be effective in a clinical trial for some types of COVID is dexamethasone, which is a steroid that tamps down inflammation. And so it's an interesting interplay between the immune system and the COVID disease. It's something that we're studying very closely in the lab.
Great. Along the same tangent, we have another audience question. Anything else to add? We've spoken a bit about this.
Can studying HIV help you understand other diseases like cancer? Yes. Wait, let me think about that for a minute. Dave, do you have thoughts?
Sure.
That's a tricky one. Well,
I mean, there's a really interesting, I think the HPV story is a really interesting take on that. So HIV doesn't typically cause cancer.
Or HTLV, Jeff.
Or
So,
HTLV, yeah.
A cousin of HIV, HTLV, is known to cause cancer.
Right.
And so I guess that's one example, isn't it?
Well, viruses that cause cancer are really interesting because you can use them. So another study that came out of Nevin's lab was studying human papillomavirus, which can cause cervical cancer, head and neck cancer, anal cancer. And what they found was that you can use the virus to point you towards the proteins that the virus hijacks in order to cause cancer. And then you can find that those same proteins, or the genes that encode them, are also mutated in many cancers. So in some cases, the virus screws around with a protein and causes cancer. In other cases, there's just a natural mutation that occurs that causes cancer. And so the virus can tell you what those genes are, so you can find them when a person doesn't have the virus. Does that make sense?
It definitely makes sense. And
I
so
think that's a good attempt to answer that question.
in terms of understanding the molecular underpinnings of how life functions, viruses can be a really interesting way to kind of wedge in there. Because what they'll do is they'll rewire everything. So what we end up doing to understand the way a virus infects cells, sometimes we end up understanding both the healthy situation and the disease situation. And so I think that's one example from the basic research perspective, illustrating that, yeah, if you study basic science, there's no telling where you'll end up or what interesting biology you'll uncover.
I have a colleague, Ben Hurley, and he has this quote that I love. He says, "Viruses are the best cell biologists."
Yeah.
It's true. We use
a lot of people say viruses know more about us than we know about them.
Yeah, I studied HIV, and I ended up, going down this path where I ended up studying DNA damage, which is involved in cancer. So you never really know where basic science is going to lead you.
It's true. HIV, because it integrates into our genome, so that has to get into the nucleus. It has to play with our genes. And so, yeah, there are definitely parallels there between that and the DNA processes that might be mutated in a cancer cell.
Great, guys. So to switch gears a little bit, I want us to talk about the social issues that outbreaks emphasize. So do you think that pandemics, epidemics like SARS-CoV-2, like HIV, bring to light the underlying inequalities in healthcare systems? And if so, what can be done to address this?
Well, I think that access to treatment, to testing is extremely important right now in terms of how to keep this outbreak under control, and making sure that it's widespread access to that is extremely important. In terms of numbers, there are certain segments of society which are disadvantaged and also seem to have predisposition towards COVID disease or having more of it. So I think to that extent, yes, it's important to make sure there's access to healthcare, most definitely.
Yeah, I think this pandemic has really laid bare the vast problems with our healthcare system. People of color are far disproportionately affected by COVID-19, especially here in New York City. There are so many factors that go into this. They are more likely to have jobs that don't have paid sick leave. They're more likely to have jobs that are essential workers. They're more likely to live in more densely populated areas. They're less likely to have health insurance, more likely to be unemployed. It also laid bare this connection between, most of us get our health care from our employers. Well, when unemployment is skyrocketing, what does that mean? What does that mean for treatment? People are sick. So I think it's a scary time. It's scary to see the news every day and see all of the problems in the world today sort of coming all at once. I think the optimist's way of looking at the situation is it's helping us identify all of the flaws in our healthcare system so that we can elect someone who can fix them in the next election.
Right. And kind of on that note, do you think that science and activism go hand in hand in the fight against HIV and AIDS?
Absolutely. I think HIV and AIDS had a really strong active component. There's the ACT UP organization in New York is one of the more famous ones, and they actually brought about a lot of change in how drugs are approved by the FDA. They really pushed through they had like a peaceful protest where they took over the FDA, and they had very specific demands for just getting rapid access to experimental treatments so that they could do these clinical trials. And one thing that HIV changed about how we do clinical trials is when there's a fatal disease, you don't give a clinical trial compared to a placebo if you know that there's a treatment that's effective. And so that changes how we do clinical trials for cancer. You always have to compare to the best treatment that's available. Before HIV, you could have somebody with a deadly disease, they enroll in a clinical trial, and you give them a sugar pill. That's not fair or ethical. So yeah, I think, like we said before, these diseases, pandemics, they expose the most vulnerable parts of our society. And really, it's up to us as a society to make noise, to say, this is not okay. We're not okay with this. We need to address these disparities and make a better world for everybody.
Absolutely. And as Jeff mentioned before, you really have to go towards the policymakers and the people who are going to fund this and get them to embrace the mission of curing these diseases, cure HIV, cure COVID-19, and make sure that we have the financial resources. Of course, with COVID, we certainly do these days, but to make sure that there are financial resources available to incentivize this work, and to make sure that we in the lab and other clinicians and scientists and other important folks combating these diseases on the front lines have the resources they need. I'm very happy to say at UCSF, I know we certainly we have the resources we need at this point to do at least the work we planned, but there's always more to be done. And so I think that activism is very important towards identifying these resources and making sure they're available for the work to be done.
Oh, right on. So as we begin to conclude this conversation, I want us to emphasize what still needs to be done to advance HIV research and care. So it sounds like we need global solidarity, making sure to bring HIV treatment to all who need it, uniting governments and policymakers to work towards a common effort, and holding everybody involved accountable. And also combining science and activism to push the narrative forward. So do you guys have any final thoughts?
I think for HIV, I work in a virology department, and there's only 2 of us out of about 20 who study HIV. And some virologists will say, oh, HIV, that's a solved problem. And I think it's important to keep in mind that a lot of people still suffer from HIV. And also living with HIV is not a walk in the park. There's elevated inflammation. There are still infections that people living with HIV get that most other people don't. There's negative consequences. And so it's important for us to remember that there are still, hundreds of thousands of people who die of HIV every year. We need to keep fighting for them and not assume that we're all good. We can do something else now.
Absolutely.
Absolutely right. Yeah. There's something like 38 million people infected with HIV in the world. And it's very important that they have access to care, and that the other people who may be living with HIV but don't know it, which a large portion of that, that they have access to testing. It's for HIV, because we don't have a vaccine, because we don't have a cure. It's so important to make sure that there's an infrastructure in place that people know they're infected, can be tested, and then can be treated to suppress their viral load. If you, under modern HIV treatment, you can suppress this viral load to a point where it can be difficult. The chance of transmission is seriously lower. And so if you can just make sure people are treated and their viral load is suppressed, then you really do a lot towards helping to stop the spread of the virus. So yes, science and activism are definitely going hand in hand, making sure that these resources are available to as many people as possible.
Mm-hmm. Yeah. So we are almost out of time, but there's one more audience question that I'd like to fit in. It's calling out the youth. So what makes you excited as a scientist or professional who studies HIV, COVID-19, And how do you hope to get young scholars involved with your chosen profession?
Yeah, I'll say I love problem solving. I like scientific mysteries, I guess you would say. What surprised me about HIV is that even though we've been studying it for, like, again, 40 years almost, it's been around, there are still entire genes. There's 9 genes in the HIV genome. You would think we know exactly what every single gene does. There's one gene that we don't know what it does. We don't know why it's there. You can delete it from the virus and it's fine in most cases, but it's still in the virus, so it needs to be there. So there's still this mystery 40 years later about what this gene does. And that's just one of many mysteries about HIV. COVID-19 is an entire mystery. It's a brand new virus. We don't know how any of it works other than comparing it to SARS, which is its closest relative. And so I think the excitement is that, as a scientist, you're looking at data or results or, scientific concepts that no one has seen before. You're coming up with truly novel information. You never know where that's going to lead. It may lead to a discovery in cancer completely unexpectedly, or it may lead to a drug or a vaccine if you're very lucky. But in any case, there's just excitement in the discovery of science, in finding things that weren't known before. That's what I like.
When we did the first interactome for SARS-CoV-2, which causes COVID-19, and I remember the day when the first data came off the machines, and some of the first interactions we saw between SARS-CoV-2 and human proteins, and as Jeff alluded to, SARS-2, which causes COVID, is very similar to SARS-1 on the genetic level, on the protein level. So we had some idea for what was going on based on SARS is 17 years old. But no one had ever done this with SARS-1 either. So we were peering into this completely new arena where there was an enormous number of interactions, molecular interactions that no one had seen before. And we were so just captured by this. An entire team of us just poured over the data, split it up, and made an entire slide deck of each viral protein. There's something like 30 of them in SARS-2. It's bigger than HIV on a genetic level. But we just looked at each one of them and explored the biology and tried to understand on a scientific basis, because you're looking at hundreds of interactions. It's such a tragic and frightening situation in the world today. But at least as a virologist and as a scientist, you in terms of understanding this, I think on an intellectual level, you can derive some satisfaction from, I think, contributing to that, the understanding of these pathogens. And also, I think it's very interesting to kind of have this glimpse into it. I think we've been privileged to have this role. I've been privileged to have that role for this particular outbreak, to help to map these interactions with an entire team of people. But as Jeff said, I think that young folks who want to enter the field of virology or any sort of scientific discipline can kind of, I hope that story gives them some motivation to pursue this field, because this isn't the last time we'll have to. I think there's a lot of work to do, and there's a lot of more interesting questions to ask. And so that's one limited experience, but then there are others. Each scientist experiences day to day.
Sure. Think about the job security. We don't, as essential workers during a pandemic, we're in a pretty good position. So if anything, go into science because you have a job.
Yeah. I feel like we could go on and on about these conversations. But we are out of time. So as we wrap up our time together, I'd like to thank you both for being part of this discussion.
Thank you.
And sharing your valuable insight, but also for the work that you do on HIV. And thank you as well to our viewers for joining us. We welcome you to subscribe to QBI TV while you're here, to stay tuned for our next episode and more. So see you next time.
Bye. Nice talking to you.